I've got 50 minutes to answer your questions or help you in whatever way I can help all of you study for the exam, prepare for the exam. And then there's office hours as usual, 12 to 1:30 today. If you've got a question, please ask. Or as I suggested up here, you can also, if you don't want to raise your hand using that old-fashioned analog technology, you can use your tablets and ask questions too. Yeah. Norm of reaction plots. Somehow I'm not surprised that that came up. Okay. So what is a norm of reaction plot? What is the question we're trying to ask?
If it's nurture or
Is it nature or is it nurture? That is the trait that we're studying controlled by genes or by environment or both? And the typical plot, well, how many axes will it have? What does a normal reaction plot look like? Yeah, it looks like an XY plot. So what do we plot on each axis?
the environmental condition.
So we've got some sort of environmental condition. So what we're going to do is we're going to take a bunch of different organisms, groups of organisms, populations, individuals, species, put them at different environmental conditions. So let's say we've got 3 different environmental conditions. Say altitude, for example. So low altitude, medium altitude, high altitude. What's on the y-axis then? Right, some sort of quantitative measurement of a phenotype. So what would we measure that might have something to do with altitude?
Size.
Pardon?
Size.
Okay, size. So let's say height. Okay. So what is it going to look like? Let's say we've got 2 different populations. Population 1 in red, population 2 in blue. So what will it look like if we have a trait, height, that's totally genetically controlled? Altitude has no effect on how tall you are.
All elevations.
Okay, so if you say it's tall at low elevation, then it would also be the same phenotype at every elevation, regardless of where you put the trait, the plant, or animal, or whatever it happens to be. I'm thinking plant for some reason. Right. This particular environmental condition has no effect on that trait. So that suggests that this is a genetically controlled trait. But what's the caveat? It's only for the condition we've studied. So we can conclude that elevation has, or altitude, has no effect on this trait. So that eliminates one possible environmental condition. So we're moving towards the idea of, well, okay, maybe it's genetically controlled. What else would we have to do to ask whether or not plant height is purely genetically controlled? We'd have to rule out every other possible environmental condition that might have an effect on plant height. So you might also have to ask, well, at those 3 altitudes, did the soil conditions change? Did the amount of water change? Did the amount of nutrients change? Etc. So hopefully you're getting the idea that it's hard to use a normal reaction plot to prove that a trait is entirely genetically controlled. What does it look like if the trait is controlled, at least in part, by environment? You start out with a plant that's tall at low elevation. What should we see? Should be some sort of change. You can't say what it's going to be, but you might see a plot that looks like this. Okay. Certainly, now, this is where we can make a positive correlation. Certainly there is an effect of elevation or altitude on plant height. So the overall conclusion is what?
Not entirely genetic.
Right, at least it's not entirely genetic. So you can never use a normal reaction plot to prove that genes only control a phenotype you're looking at, but you can use it to prove that there's at least some environmental effect. So a line with slope of 0 or something close to it, gray area here, suggests that there's no environmental effect. If there's a slope, that suggests there's an environmental effect on the trait. And it's not always going to be a straight line. Sometimes you might see things that might be confusing to interpret. Like you might look at a third strain that has some sort of strange pattern, no consistent trend. That's fine. That just says that environment does affect the trait. So I'm going to put a big question mark there by, is it genetically controlled? Can't say for sure. We've just ruled out for the blue trend line. We've just ruled out that this one particular environmental condition doesn't affect the trait. Bless you. Any follow-up questions on normal reaction plots, additional questions, concerns, want another example? Yeah.
I was just wondering, I remember something about like overlapping lines indicating something.
Sounds like something I put into class. I'm having this flashback. That sounds familiar. So overlapping lines.
You said it was environment, if it was overlapping.
So it suggests if you have multiple, so it's mostly about the slope. That if you change the environment and the phenotype changes, then there's a relationship between the environment and the trait. That part of that lecture that you're referring to, I believe, refers to the idea of comparing two different individuals from the same species or two populations from the same species. And if you identify, if they look similar, so if instead black had looked like that series, looks exactly like the blue. If these are two genetically distinct, so if this is genotype B, so we know that there are genetic differences between these plants or animals, whatever they are. This is genotype A, different population, same species, they're genetically distinct. If they have the same reaction to environmental change, that also suggests that their genetic differences, those are two genetically different strains, but they have the same environmental response. Also suggests that genetics aren't involved. The genetic difference between the strains or populations doesn't affect their response to the environment. I would be happy to repeat that. So in the case of these two lines where you have two genetically different individuals or populations, there's a genetic difference between them. So a hypothesis might be that that genetic difference, say genotype B has an A at one position in one gene, sorry, genotype B has an A. Genotype A has, say, a C at the same position. So you know there's a nucleotide difference. You hypothesize that that affects their ability to grow at different climates. And you compare them and you see that the lines are exactly the same, the two trend lines. That implies that that genetic difference between those two populations doesn't affect the trait that you're measuring. It doesn't matter if you've got an A or a C at that spot in that gene. You have exactly the same response to change in that environmental condition. So that's one way to rule out genetic control of a trait. Okay. So, synapomorphy, symplesiomorphy, and homoplasy. It's magic. The system works. Okay. So imagine that you have whatever this is called, phylogenetic tree, 3 lineages are related to a common ancestor. Is this a useful phylogenetic tree or not so useful? It's not as useful. It doesn't tell us anything about how these populations or species are related to each other. So what would we like this tree to look like? If you have 3 species, what would an optimal tree look like? Yeah, so you'd hope to find something that looks like A and B are more closely related to C. So what's a trait that A and B share in common? Who knows? Let's say this is a plant and they have yellow flowers. And outgroup now in this tree on the right, C has, I don't know, white flowers. So if you map these traits on the tree, on the left, A has yellow, B has yellow, C has white. W. So what is the fact that in this polytomy on the left, A and B share the same trait? What does that do to those branches when we draw the tree on the right? It brings those branches together because those 2 populations share a trait. It synapses those branches. So you start with the polytomy, but when organisms share a trait, that closes those branches together and produces the tree that we drew on the right. So synapomorphies are the ones that show us the close relationships between species. So synapomorphy is the trait that organisms share because they have derived it from a common ancestor. These are the traits we like. Bless you. Yeah.
I have a question about that, because doesn't everything come from one common ancestor? So where do you make that distinction?
Okay. So do all traits derive from a common ancestor? Well, depends, in my opinion. So DNA, for example, we discussed, right, probably came from the original ancestor of all living things, right? It's a trait that all living things share. So that's an example of a trait that, yes, probably the only trait, maybe also the requirement that you ingest nutrients, that you reproduce. Like there are a few traits that are common to every living thing on the planet. Other than that, what's a situation in which you would not have a synapomorphy, but you would have traits that organisms share not because of their common history?
Environmental effects that can cause them to kind of evolve the same type of traits based on their environment.
So a pressure that causes the evolution of the same trait in 2 different places. Okay, so that might be the light sensing proteins in C. elegans, the worm I study, and the fact that we have eyes. So both worms and humans can sense ultraviolet light. We sense it by sunburning. They sense it by having a protein that actually responds to ultraviolet light. So is that because worms and humans have a common ancestor that had the ability to see light? No. In that case, worms and humans evolved a similar trait, eyes, on the branch that get humans, and more recently in time, UV light detection in worms. So if we just surveyed all the living things and we asked who can detect ultraviolet light, some fish do. Some fish have eyes that can actually see in the ultraviolet. Butterflies can. You might have seen those cool pictures of flowers taken under ultraviolet light. And you can see all these new patterns emerge that we can't see because we don't see in the ultraviolet. But butterflies can see these patterns. It's crazy. Let's Google it right now if you want. So all of these traits evolved independently. So these don't suggest, so parallel evolution, the separate evolution of different traits in different lineages is not a synapomorphy, it's a, the other one, homoplasy. Symplesiomorphy is the third one we're going to do. So this is the most simple comparison, synapomorphy and homoplasy. Synapomorphy is a shared derived trait. It tells us information about the relationships between species. Homoplasy is equivalent to independent evolution of the same trait. So these are the types of traits where you would study an organism and you'd think, oh, they share the trait, they must be closely related, when they're actually not. It's misleading information. Does anybody have a better example of homoplasy than ultraviolet light detection?
I think in the book it talks about like the mouse coat color and then also the lizard coat color, kind of like where they live.
Fair enough.
Or
Yeah,
coat color.
right. So that's another example of parallel evolution. You have mice that live on beaches evolved a light coat color. They live in rocky, say, volcanic habitats. They have a dark coat color. The same is true for lizards. The lizards that live in light environments, some lizards, have evolved light skin, and the ones that live in dark environments evolved dark skin. That doesn't mean that lizards and mice are sister species. It just means they happened to have evolved the same trait. Yeah.
That confuses me. What's the difference between homoplasy and convergent evolution?
They're the same.
They're the same.
Yeah, homoplasy is convergent or parallel evolution, the independent evolution of two traits in two separate branches of the tree of life. Okay, now symplesiomorphy. It's not so simple. Okay, so does anybody remember what a symplesiomorphy is, or have a brief definition of symplesiomorphy?
It was present in the common ancestor,
So it's a trait that's present in the common ancestor.
but some of them lost it.
Okay, so I'll just say that these organisms all have a blue plus on their bodies. It's a trait you can measure. Okay, so what would this tree look like if this is a symplesiomorphy? This trait is lost somewhere on this tree. So let's say right there. Which organisms from top to bottom, let's name the species A, B, C, D, E, F. Who's got this trait and who doesn't? Right. D, E, and F share this trait because it was passed down from the common ancestor of all 6 of these species. And then A, B, and C don't have this trait. They don't exhibit blue pluses on their bodies. Some weird evolutionary tattoo or something. Okay. So the problem with the symplesiomorphy is that when you see D, E, and F share this trait, you wouldn't necessarily draw a tree that looks like this. You might instead group them together because they share that trait. So you might be tempted, for example, to draw the tree like that, indicating that D, E, and F have a more recent common ancestor. And this would, by the way, would be a parsimony tree. So you would maybe in this case argue that plus wasn't ancestral, that it evolved on this branch. So the original example was what I'm saying if we went back in time was actually true, that the common ancestor did have this trait, and that it was lost in the lineage leading to A, B, and C. But with the Symplesiomorphy, when there's an ancestral loss of a trait, that can affect how we might guess, hypothesize the populations are actually related to each other. So of course, this is kind of intellectually unsatisfying because we can never go back in time and find out what really happened. The point is simply that if you find out that a trait is a Symplesiomorphy, you might guess that you've drawn the wrong tree. Most of the time we can't know if a trait's a Symplesiomorphy. We just have to try to avoid them. So avoid Symplesiomorphies, focus on Synapomorphies. Symplesiomorphy, loss of an ancestral trait in one part of a tree can give the impression that those species are more closely related to each other than they actually are. So misleading trait. Other questions? Follow-up questions?
If
Oh,
one tree has
yes.
two sister species, is there a special name for that?
Can you say that again, please?
If one tree has two sister species like B and C and E and F, and this.
So to draw it most simply, if there's an outgroup and then there's like this. Actually, there's no outgroup there. There's an outgroup. So is there a special name for a tree that has two pairs of sister species? Not particularly. There are lots of trees have this sort of structure. In fact, let's see. We can always add, and this is a discussion we had sort of towards the beginning part of the phylogenetics exercise. What is shown at the branch tips?
Living species.
Okay, living species. What would it look like if there was an extinction on this tree? So this is present time. So what would it look like if I represented an extinction event?
They didn't touch the present line, it's vanished.
Yeah. So any extinction would represent organisms that don't exist at present. Okay. What isn't on this phylogenetic tree? We've got 1, 2, 3, 4, 5, 6 living species represented.
There's no
I mean, how many species are there on the planet? I don't want you to answer. I don't know. Google it. Wikipedia, maybe. Right, so we're not representing lots and lots and lots and lots and lots of branch tips. So this tree could also be drawn with that sister species. And there's actually, if I zoomed in and started drawing all the species, we would just get finer and finer resolution. But there would always be lots and lots and lots of sister taxa on the tree. We just choose which species to represent and which not. And as far as I know, there isn't a specific term for a tree that just has two pairs of sister taxa. Let's see. James Bond wants to know how we measure frequency of alleles over time. Nice one, 007. We haven't gotten there yet, population genetics chapter, but that's what we're about to do. So if you really want me to answer, tell me on today's meet. But that's not going to be on the exam. So in general, though, briefly, to sate the curiosity, hopefully, measure allele frequencies. You actually go out into nature, you collect individuals, you extract their DNA, and you somehow read, genotype their DNA. So you go out and you pick obviously you can't take all of the individuals or else you'd be a bad biologist. Shame on you. You take a sample of individuals, you look at their DNA sequences, and you actually look at how many of each allele at a particular gene is present. We'll get into more detail when we get into population genetics. Let's see.
Oh, I was wondering if you could quickly go over parsimony, just.
Okay, so the concept of pony, then we'll work on redrawing phylogenetic trees in different ways. Okay. So what are the two different approaches we discussed drawing trees with? There was parsimony, and there was distance. Yeah, not to be confused with maximum likelihood, which is something we didn't talk about. If you really like phylogenetics, take Dr. Crosby's systematics class. Everybody's a fan? Okay, good. We've got distance and we've got parsimony. So in distance, you do what we've been doing the last few days in class. You take, for example, this is not the only way to do a distance tree. You take DNA sequences, you align them, you count the number of differences between different individuals or different species. Something like that. You construct the distance matrix comparing A with B, A with C, and B with C, which I won't do explicitly unless you want me to. So you don't fill in that part of the table. You fill that in, and then you use those distances to draw a tree with branch lengths. The approach to drawing a parsimony tree is vastly different. So you take some collection of information, like, let's make something up. You have, what's a trait we can measure? Tail, presence or absence, and number of limbs. Two or four. I don't know, something like that. For those species, you could collect, let's say 5 species. You could collect that information, tail, number of limbs, so it turns out that species A and D have tails, the rest don't. And in terms of number of limbs, who knows? Let's see. Something like that. So this is a sort of a de novo approach. You try to create a tree that explains this distribution of traits with the fewest number of transitions, changes, gains or losses of traits. So with no more information. So how would we go about doing this? Did I do that? I didn't mean to make those two columns the same. That doesn't matter. So which species are most closely related to each other? Let's start there. So make myself a little bit more room on here. So B and C are close relatives. That's a C. And
A and B are more close.
B, C, and E all have the same pair of traits. They don't have tails and they have 4 limbs. And then A and D. Okay. Nobody threw something at me. So here's one tree. Is it a parsimony tree? So so A and D are sister species or taxa. B, C, and E represent a polytomy. That's fine. But if you drew on this tree where gains or losses, there's no root, which is fine. Where on this tree did a transition from 2 limbs to 4 limbs happen?
Going towards B, C, and E, they got 4 limbs.
Yeah, so somewhere here in the middle there was a transition. We don't know which direction, if it was from 4 limbs to 2 limbs or 2 limbs to 4 limbs. We would have to have what on this tree to know the direction of evolution? That's what, yeah, we'd have to have some representation of the ancestor. We'd have to have a rooted tree. Where did the transition from tails to no tails happen?
Same spot.
Well, yeah, the same tails plus to tails minus. We don't know whether or not these happened at the same time or not. We don't know whether these differences or similarities in traits are related to each other, but they look like they happened at the same spot on the tree. So in terms of parsimony approaches, this means there were 2 changes necessary on this tree to represent all of the data that we have in our tree. What would be a less parsimonious? This is almost it's almost ridiculous for me to ask you this, but what would be a less parsimonious tree? It should hurt your brain to have to come up with a less parsimonious tree than this. So, okay, I think the relationships between species look like this. So that's a hypothesis. It's definitely, I can already tell, less parsimonious.
Which one's
There's more changes that have to happen.
Oh, I'm sorry. Good catch. Let's call that E. Okay. So if we map all these traits onto this tree, B is n and 4, C is n and 4. So that's okay. They share a trait, they're closely related to each other on the tree, but where else is there an n and 4?
A.
E is n and 4. D and A are Y and 2. Yes, they have tails, they have 2 limbs.
Could you safely say that E is an outgroup of D and A?
Well, the question is, when dealing with parsimony trees, which was the original question, where would you have to now draw the changes on this tree to represent what's going on?
First, if they lost limbs, going towards D and A.
So there's
Now they gained it again, going towards E.
right. So there's a 2 to 4 transition in there somewhere. We have organisms B and C that have 4 limbs, DNA that have 2, but that means there must be some sort of a change again in that trait right there. And then what about tails plus or minus? It's the same. So there'd also have to be an independent gain or loss of tails or no tails. So that requires 3 changes on this tree to place all of the data on. So that's less parsimonious than the first tree. So the first tree is the most likely, the most parsimonious explanation of the patterns of data that we have. Okay, so quick follow-up questions on this one, and we'll start redrawing some trees. Today's meet is clamoring for tree redrawing, and no one's tweeting, which is fine. I think. Okay. So here's a tree. Let's do a bunch of fun stuff. Which are the most closely related species? Trick question. So A and B and D and E in this case are both sister species.
Branch lengths for A and B are shorter.
So in this representation of a phylogenetic tree, the branch lengths, well, I meant them to be the same. Here, let me, where's my eraser on this thing? I meant them to be the same. So if, by the way, on the exam you feel like you want to make that sort of a point, that's totally fine. Write down on the question any assumptions you make. Like, it looks to me like the branch lengths are longer the way you drew them, therefore I'm assuming that those species are more distantly related to each other. That's totally fine. I'll grade your exam based on not only what I think the correct answer is, but what assumptions you tell me you made when you answered the question, if you feel like you need to make an assumption. So in this case, I meant for A and B and D and E to be both sister species that are equally related, A and B to each other, that is, to D and E. Who's more closely related to C?
D and E.
D or E, okay. So it's both D and E are equally related to species C. How would we redraw this tree to put C on top?
You bring A and B there.
Right, so you could just swap those two sets of branches. It's basically rotating the tree right there. So taking the whole tree and rotating it. So what would that look like? We put A and B down here. We put C, D, and E up there. So do those two trees represent the same topology, the same shape, the same relationships between all of the species, or did I screw up? So are D and E and A and B still sister taxa?
Yes.
Is C still the next most closely related species to D and E in both trees? And they all share a common ancestor. So they're the same tree.
Isn't C farther away from A and B now?
Ah, so that's the critical question is, does the distance between the tips, the labels of the branches, tell us anything about the relationships between the species?
You can switch A and B though. Can we?
Sure, you could redraw this tree by rotating there.
So now they are equal.
It's still the same tree. You've just relabeled the branches. So it's not relationships between the species, how closely or distantly related they are, has nothing to do with the order of the labels on the tips. What's the unit? When you talk about how closely related you are, think about your family tree. Who are you most closely related to? Your parents or your grandparents? Your parents. Are you more closely related to, oh, I shouldn't go any farther than that. I'd have to describe what the difference between first and second cousins and the degrees removed is, so we won't go there. So the point is, simply, the unit of measurement we use to describe how closely related organisms are is time. The time it takes, how far back in time we have to go to reach a common ancestor. So we're more closely related to our parents than our grandparents. So the same is true here. So in all of these trees, when present is the branch tips, in both of these trees, we have to go back to where to find the common ancestor between, say, E, A, and B. So E, we go back in time to basically the root, which is where we find the common ancestor of A and B. So does that degree of relationship between E, A, and B change when we redraw the tree like this? No, you still have to go to that same spot to find the common ancestor between E and A and B. And the same is true up here. So here A, B, and C are written in that order on this tree, but you still have to go back to the common ancestor of all 5 species to find where A, B, and C share that common ancestor. And that doesn't change when you rotate nodes. It's still that point in the tree, that far back in time, same distance to find the common ancestor. So really, you can rotate branches and at nodes all you want. You can change the order in which the species are listed, but that order that they're listed in has nothing to do with their relationships. It's arbitrary, essentially, as we saw when we rotated this branch here on the bottom and flipped the positions of A and B. We choose which order we put sister taxa in. Who's on top, who's on bottom, who's on the left, who's on the right. So I don't know if this is helpful. Think of it as a baby mobile, right? The thing that hangs over the crib and spins. Like that's what a phylogenetic tree is. You could, if you have a really good imagination, you can imagine grabbing it at the root and dangling it and just letting all of these branches spin. Right? You can twist things. It doesn't change the relationships between the positions of all the branches. It just changes where in the room or where on the tree the labels are. Don't make me bring in my pipe cleaners. There's a fabulous pipe cleaner exercise we can do with building phylogenies, but we're almost past that part of the class now, I think. We're on to 2nd grade.
I have a question about the study guide. Will there, for number 9, that was a little complicated and time-consuming. Will the test have something similar?
So there's a concern about the amount of time it takes to do certain questions. Let me check. And if anybody has a follow-up question to this, think about that while I'm checking out question 9. Okay, so question 9 is horrible. 6 species and 15 nucleotide sequences. How long did it take to answer that question? More than it should? Okay. More than you'd want to spend on one question on a 50-minute exam, maybe? Okay. So, no, I would not normally put a question that would take that long to solve on an exam. And if I did, the point value would be weighted accordingly. That is, it would be worth like a point. So this is my philosophy on exams. So you all know that the grade distribution is 20, 40, 60, 80, 100. So you have to get 4 out of 5 points on average to get an A, 3 out of 5 to get a B, 2 out of 5 to get a C. So I do write, as every professor does, some questions that distinguishes A students from B students. There will be maybe one really hard question on the exam that I don't expect a lot of people to get, but that's not worth a whole letter grade's worth of points. So there will be a hard question on the exam. And if you have time to finish it, then good job.
How many questions are there?
Which really doesn't tell you anything, but I will happily answer that question. Because since you've never seen in one of my exams, I don't know if it matters how many there are, but let's see. There are what I call 8 questions.
All multiple choice or how is it?
And that's about all I can tell you. So it's 8 questions, it's 20 points. So you can figure out approximately how many questions per point, and one of those questions is probably more difficult than the rest. And as I suggested on the syllabus, which I suggest you look at before the exam, look at the grading rubric. What I say it takes to get, what do I expect from you to get 20% or 40% or 60%? So what I try to do is aim towards, I don't want to take up a ton of your time. You should be able to get at least 20% by having shown up to class and memorized factual information. So maybe about 20% of the exam would be multiple choice testing factual recall. And because you all have your tablets and access to your notes in the internet in class, I expect nobody to fail. To get between 0 and 20%. At the very least, you can spend 50 minutes looking up the answer to one multiple choice question. Everyone's graduating. And then it goes up from there. So to get the last 20%, the top 20% of points, as it says on the syllabus in the grading scheme, I describe what it takes to get a D, a C, a B, and an A. To get an A, I expect you to be able to integrate information that you've learned in class to do something maybe new that we've not seen before, or at least to combine different parts of information that we've studied during the semester so far in a synergistic, synthetic sort of way.
Do you do partial credit?
I do partial credit. I do assign partial credit. Yeah, also part of my grading strategy. And that's why I suggest showing as much work as you can on an exam. That helps you figure out at what point did you stop understanding the concepts. And yes, so partial credit, absolutely. With the rare exception of multiple choice. Sometimes on multiple choice I can't do partial credit. Other sorts of questions, which is the vast majority of the exam, yeah. Other questions? Content or about the exam?
So there will be some multiple choice questions, is that what you're saying?
Yes. The multiple, I don't know. I don't remember the exam already. I wrote it a week ago. It's likely that there will be multiple choice questions, but they will require you to do more than just memorize facts. So it will require you to do something and then pick the right answer. So here's a distance matrix, which is the correct tree. That sort of thing. So I'm trying to save you a little bit of work by providing possible answers, but it will still require more than factual recall to get the correct multiple choice response. 1 more minute.
It's all individual, right? It's not a group.
Yeah, right. There is, for those of you that had me with genetics last semester, there are no group, at least on this one, we'll see. Midterm, no group portion of the exams. It's all individual. Sorry for those of you that like group exams, and congratulations for those of you that don't. All right. Well, thanks for joining in. I'll post this video as